All right, hello, this is Christina Howard. This is a presentation for Biology 241, and this is going to concern chapter 10 of the Martini textbook, which is muscle tissue. So without further ado, let's get started. We're going to talk about mechanics, so how they get shorter and longer, and link that to physiology. So what's happening at the cellular and microscopic level that makes your muscles contract from front to back. And unfortunately, my computer is currently charging, so I can't turn it into tablet mode at this time, which means I'm going to have to limit my drawings. But once it is charged up enough, I'll just go ahead and pause recording and put it in tablet mode so I can do some more drawings. So first of all, let's discuss the kinds of muscle tissue. And fortunately, uh, this series of lectures is going to focus primarily on skeletal muscle because the model by which it contracts is basically the same as cardiac muscle. Um, and so talking about things like sarcomeres and things like that will apply to both kinds of striated muscle tissue, so cardiac and skeletal. And then we will talk a little bit about smooth muscle, but it won't be the primary focus of our discussions. It'll mostly be striated muscle, so again, that's skeletal muscle and cardiac muscle. So with regard to skeletal muscle, obviously voluntary movement is one important function. So that's how I'm talking to you. That's how we can walk around, move around the environment we live in, manipulate objects, etc. Another function is to maintain your position or posture. So if you're sitting or standing right now, you're probably engaging your core muscles and your neck muscles to keep your trunk and your head upright instead of flopping over to the side. Supporting soft tissue is another aspect. So your abdominal cavity is guarded by some pretty sturdy, multi-layered muscles that form your trunk, so your abdominal muscles and your back muscles. And this helps to support that soft tissue as well. And you have a variety of skeletal muscle sphincters which guard openings. So you'll learn more about these next term. But for example, both the urinary system and the digestive system have external sphincters, which are skeletal muscle, and these are the voluntary components of urination and defecation. And then they have internal sphincters as well, which are smooth, and those are the ones you don't get to control. Help maintain body temperature. This one is related to shivering. So that's the rapid contraction and relaxation of muscle tissue in response to cold. And that just generates heat by friction. And then finally, a stored nutrient reserve. So there is glycogen stored in our skeletal muscles, and worst case scenario, we can begin to break down our muscle tissue to use it for fuel, but that's like the bottom of the priority list. So your body will run through all the other fuel it has available first before it turns to muscle tissue. So with regard to the organization of skeletal muscle, let's take a quick look. So just to give you some context here, this is going from the smallest level. So we have a muscle fiber, and you'll note that parentheses it says cell. So for skeletal muscle, we are using the word fiber interchangeably with the word cell. And that's because skeletal muscle cells are long and fibrous. So an individual cell can be very, very long in this particular tissue type. And then as we go up this little illustration here, we'll see we're getting farther and farther zoomed out, so larger and larger organizations. The other thing you'll notice is that 3 of these terms end in the word mesium. So MYS is the root word that refers to muscle, and epi, or peri, or endo refers to the level of structural organization. So at the smallest level, each muscle fiber has of course, its sarcolemma, which is the cell membrane of the skeletal muscle cell. So sarco means flesh, and this prefix is one that we append to any skeletal muscle cell anatomy thingy. So you'll notice sarcolemma, sarcoplasm, etc. So sarcolemma is just the cell membrane, but on the outside of that, lining the cell itself, is the endomesium. So each individual muscle cell gets its own liner. And this is a place where tiny capillaries are found. So these are the capillaries that directly supply your skeletal muscle. So this is going to be a thin layer of connective tissue that just wraps around each cell. One level up, a bundle of cells So we have multiple muscle cells packaged together. They're each going to have their own endomysium, but all of them are going to be surrounded in a packet by paramysium, peri meaning around. So this is going to surround multiple bundled up skeletal muscle cells. And then finally, the entire muscle organ, so consisting of multiple fascicles, the fascicle is the name for the bundle of muscle cells surrounded by paramysium, multiple fascicles are surrounded themselves by epimysium, which is again connective tissue wrapping that wraps multiple fascicles. And so what you can see is that there's a sophisticated and very fancy system of linings and wrappings all the way from the smallest level of structural organization up to the largest. And this is going to help isolate your muscle tissue from neighboring muscles. So that prevents your action potentials from spreading where they shouldn't spread. It's also going to help prevent against infection traveling. So if you get an infection in your skin, it's harder for it to get into the muscle unless the epimysium is breached for some reason. And when the muscles are tightly packaged, it helps to minimize the lateral sort of bunching of the muscle so that the contraction is maximally efficient at shortening. So they're tightly packaged for that reason as well. So your skeletal muscle gets a lot of your blood supply. So if you were operating under the assumption that all of your body organs and tissues get the same amount of blood, it may surprise you to learn that that's not actually true. So there are cells and tissues that are very greedy for oxygen and nutrients because they have a high metabolism. Nervous tissue and muscle tissue are pretty high up there. So additionally, it's going to depend on, how active you are. So if you are just sitting in a chair talking like I am, that's pretty equivalent to resting. So per minute, my skeletal muscle, even at rest, is getting around 1,200 milliliters per minute of my cardiac output. So that's pretty high. If I were to get up and go run around my house a couple times, that number would rise to between 4.5 liters, that's how many milliliters makes up 4.5 liters, 4,500, up to 12.5 liters per minute during activity. So if I'm really working hard, I can approach 10 or more milliliters per minute of my blood pumping out to my skeletal muscles because they just really need that oxygen if I'm going to place that kind of exercise demand on them. So regarding the nervous supply to muscle, what you're seeing here is the spinal cord and reaching away from it, a variety of spinal nerves. You can also see the cervical plexus here, which is going to be, you'll learn all the plexuses later. I'm just kind of showing you them because they're here and it makes sense to show you what controls which things. So you can see that the cervical plexus is going to innervate your neck muscles and your trapezius. Your brachial plexus, which is this group of nerves, is going to innervate the muscles of your arms. And then your intercostals and to some extent your abdominals are going to be innervated by these nerves right here, which are the intercostal nerves. And then going further down here, we have the lumbar and sacral plexus, and these are going to be the nerves innervating the muscles of your butt, lower trunk, thigh, and leg, among other things. So we call the signal that leaves the central nervous system and goes out to the skeletal muscle an efferent signal, E-F-F-E-R-E-N-T, and that means leaving the central nervous system and acting at a distance on some effector, in this case, the skeletal muscles. The majority of skeletal muscle control is voluntary, meaning that you get to decide if you want to move your muscles or not, but as we all know from having muscles ourselves, there are some skeletal muscle-based reflexes, like what happens when you grab a hot pan or taking breaths, that are not entirely voluntary. So there is a sort of workaround switch in cases where your central nervous system needs to take over control from you for a short amount of time. So the features of skeletal muscle cells we discussed a little bit when we talked about their histology in lab, but I mentioned that one of the hallmarks of them is that they're multinucleate. So one cell is going to have a lot of nuclei, and that differentiates them from cardiac muscle and smooth muscle for whom there's only one nucleus per cell. That's kind of the usual rule, is there's one nucleus per cell. Sometimes, in certain situations, there's more than that, and this is one of those situations. So during development, you have a bunch of little cells called myoblasts. So myo meaning muscle and blast meaning precursor cell or baby cell. So mesodermal cells, which are stem cells, differentiate into myoblasts, and then the myoblasts begin fusing with each other. Once they've fused significantly large into a large multinucleate cell, then we have the growth of that cell with multiple nuclei until we get this mature muscle fiber. So an immature fiber doesn't have the sarcomeres yet, which are these orderly bands of proteins, but it will. It just has to make them, and the nuclei, of course, are going to be responsible for that, along with the endoplasmic reticulum, because of course that's where the transcription and translation of proteins happens. So during development, all of the proteins of your muscles are being made. Once those fibers are complete, we have this internal protein arrangement called sarcomeres, and I'll show you the details of those in a minute, that result in visible striations like the ones that you observed in lab. So, the details of a fiber. So we have a larger zoomed out view of a fiber right here, and then in here, we have an inner view. Now, this is a cartoon, obviously, so these structures aren't really blue and yellow as the image would have you believe, but they're colored artificially just for contrast purposes, so you can tell the difference. So the sarcolemma, again, sarco means flesh, lemma means layer. So this is just the cell membrane of the skeletal muscle cell. This is going to conduct the electrical signal for contraction. So I talked about this conduction capability in the podcasts episodes that I released recently, but I'll talk about them as well in this video. And you can also look at the Khan Academy style drawing videos that I have about all this stuff to reinforce those concepts. So I will still talk about them here. I'm just telling you some other places where you can consume that content. So these are packed full of what are called myofibrils. So myo meaning muscle and fibrils meaning little tiny proteins inside of the muscle cell. So you can divide these into thin filaments and thick filaments. So they basically match their description. One is a lot thicker than the other, and I will show you in detail which is which. Now, the transverse tubules or T-tubules are going to be little holes, and let's see if it will let me zoom in here. Yeah, there we go. So what you're going to see is there's little holes in the surface of the sarcolemma, and it's not very well illustrated here, but what these are doing is tunneling into the cell in a network like this. So you can see here's the openings of some T-tubules, and then they're going down into the interior of the cell, forming a network of tunnels. And what this allows is for the impulses that are spreading across the sarcolemma, the electrical impulses, which are driven by ion flux, to descend into the cell to effectively excite all of these myofibrils. So that's why that T-tubule exists. So the transverse tubules are filled with extracellular fluid. So they're the same fluid as what's around the muscle. It just gives the inside of the muscle access to that extracellular fluid as well. And that's going to conduct that electrical signal deep into the cell. There's also the sarcoplasm, which is the fancy name for the cytoplasm of the skeletal muscle. So this has glycogen, which you might remember is just a bunch of glucoses glued together in a branching chain. It's a way to store glucose for fuel if you need it. There's also myoglobin. So myoglobin is kind of like hemoglobin. Myoglobin is a molecule that hangs on to oxygen and lets it go when there's an increase in demand for oxygen. So if you go to the store and you buy like a steak, for example, what you're going to notice is that there's a little sort of almost maxi pad-like thingy under the meat that is going to absorb the juice that comes out of the meat. And it's going to be pink. Most people assume that that juice is blood, but it's not actually blood. It's myoglobin coming out of the cut muscle tissue of the steak. So if you think about activity levels changing and skeletal muscle, what you'll notice is that the activity levels and therefore the oxygen demand of the muscle is going to change a lot over the course of a day, an hour, a month, a week, a year, depending on how active you are. So, accordingly, having this little oxygen storage molecule around just in case the activity level suddenly changes is a good way to help keep the oxygen demand meeting, or excuse me, the oxygen supply meeting the demand. Ordinarily, the sarcoplasm has low calcium, and that's because all the calcium is stored in this sarcoplasmic reticulum. So the calcium is not bopping around in the sarcoplasm, it's in this enclosed structure, which is kind of like the calcium garage. And finally, there's going to be super abundant mitochondria all over the place, packed in amongst the myofibrils and the sarcoplasmic reticula. And that's because, of course, all of this, if it's going to get shorter, is going to require a lot of ATP. And so it makes sense to have ATP factories positioned all over the inside of the cell. So in addition to having many, many nuclei, the muscle cell is also going to have many, many mitochondria. So as I mentioned, the sarcoplasmic reticulum, it's basically a specialized smooth endoplasmic reticulum. It's just specialized to store calcium specifically. And one of the proteins that it has inside of it is called calsequestrin. So to sequester something is to gather it up and store it somewhere. So calsequestrin is a protein that's basically going to gather up calcium and allow it to be stored and saved up inside of the sarcoplasmic reticulum. It also helps to gather the calcium ions back up after they're released. We'll talk about that as well. And finally, triads. So once again, if we zoom in to this picture, what you'll see is that there's a networky area of the sarcoplasmic reticulum, and then there's a uniform area without that lacy appearance at either end, here and here. So a triad is one T-tubule, illustrated here in yellow, and then the two sort of smooth, unified places of the sarcoplasmic reticulum that are illustrated here, and that's called the terminal cisterna. So terminal meaning end, and cisterna meaning cistern or storage place. So the terminal cisternae, 1, 2, is the plural way to say that. So a triad is one T-tubule and two terminal cisternae. Okay, so I have to take a pause really quick, just because if you hear the clickety-clacking near me, it's my very elderly dog wanting to get let out. He's probably not got much time left on this earth, so I'm trying to let him go outside as much as I can. So I'll be right back after I let him go outside to enjoy the grass and go potty. Be right back. All right, sorry about that. Thanks for your patience. I am back, and hopefully the clicking and clacking of the nails on the hardwood floor wasn't too loud for you. I apologize. So let's get back to it. I have mentioned in the calcium homeostasis content, among other places, that calcium is a really important molecule for a variety of reasons. I talk about that in the podcast and in other content. But here's another reminder. So for calcium to be used by your muscle, it has to be sequestered and divided into particular compartments. So relating to skeletal muscle physiology in particular, we measure that in millimoles per liter, which millimoles is 1,000th of a mole. If you're not familiar with a mole, this is a way to count the number of atoms of a substance by its molecular weight. So a millimole is 1,000th of a mole, and then per liter is per liter of fluid. So in the cytosol, which is to say the liquid inside of and between all these organelles, the concentration is 1 × 10⁻⁷ millimoles per liter. So here's what that number looks like if you actually multiply it out. This is a very, very tiny amount. And it's tiny on purpose because we want this to be low until we want to begin contracting. The extracellular fluid has a whopping, by comparison, 2 millimoles per liter. So what that means is most of the calcium that we're going to be letting in is going to come from the extracellular fluid, but a significant portion also comes from the sarcoplasmic reticulum at 10⁻⁴ millimoles per liter. So it's a couple orders of magnitude larger than this tiny little number here. Okay, So moving on to the structure of sarcomeres, and if you're like, wait, what about the calcium? What does it do? Never fear, I will explain. I just have to talk about the sarcomeres in order for anything about that calcium stuff to make sense to you. So this is the functional unit of the myofibrils and of the muscle cell itself. So without these sarcomere structures, which are made of a repeating organization of proteins, your muscle cells can't contract, specifically your striated muscle cells. They just don't go. So this is the basic unit of getting shorter that exists. So you can have as many as 10,000 sarcomeres lined up end to end per fiber. That it says may here because of course it depends on how long the muscle fiber is. So a muscle fiber in your leg, for example, one like the gracilis, which goes all the way from your groin to, or excuse me, from your outer hip to your inner knee. Or no, gracilis is groin to knee. Sorry, I was thinking of the sartorius, which is outer hip to inner knee. That has lots of really long fibers, like a couple feet, versus a very short skeletal muscle, something like the levator labii superioris, which is a short muscle that allows you to lift your upper lip. That's not going to have nearly as many sarcomeres because it's very short. So these extend from Z line to Z line. You may also see the Z line referred to as a Z disc, because really it's only a line when you take a longitudinal section of these fibers. If you were to turn this and look at the cut face of the myofibrils, it would look like a disc of proteins to which other proteins are adhered. So the A band is the length of the thick filament, and don't worry, I will zoom in on all of these and you'll also get a drawing, just I have to wait till my computer is charged up enough again. And the I band is the area between A bands. So let's just have a look at that up close. So here we have the A band, and as you can see, the thick filaments here, which are made of a protein called myosin, the length of those thick filaments is the width of the A band, and that's always going to be true. That never changes. The I band is the distance between the thick filaments, and that includes the Z line or Z disc at its center, and the thin filaments and this protein called titin, which just keeps everything in line with each other. So you'll notice that there's this area here where the thick filaments and thin filaments overlap. So this is the business end of the sarcomere. Where there's overlap is where there's tension being generated. And you'll come to understand that better with time. So if it's not quite clicking yet, don't worry, it will eventually. But because these are arranged in a repeating organization where there's one after another, we have to decide where one sarcomere ends and the other begins. And so that's been decided that it goes from Z line to Z line. And here's what that same stuff looks like in a transmission electron micrograph. So up here we have a cute little cartoon of it, and then down here we have a electron micrograph of an actual set of sarcomeres. And you can see that even though it doesn't look quite as tidy, all of the same parts exist. So we have the Z line, which is very conspicuous here. We have the M line, which is the stabilizing core of the A band. We have the A band, and then we have the I band, which is this pale part where there's only thin filaments and titin, and then their zone of overlap is here as well. So it's nice when there's proof of concept that, hey, the stuff I'm telling you about the tiny molecular arrangement really does exist in real life, and I can show you that in a picture. So I like this image in particular because what it's showing you is a sarcomere, and it's showing you the longitudinal view. And then each of these little circles is showing you what that sarcomere looks like if you cut it and looked at the cross-sectional view of that spot. So at the Z line, you can see it's really discs. And so there's an attachment point for titin, which is that green spring-like molecule, and then there's actin filaments, which are all attached to the protein of the Z line. In the middle of the I band, it's just thin filaments, which are the red guys, and titin filaments, which are the little spring stabilizers, which are the green ones, in this repeating arrangement. Right in the middle of the M line, that's the anchor point for all of the thick filaments. So you can see cross-sectional thick filaments all anchored to each other. In the H zone, which is an area right about here where there's no thin filaments yet, there's just thick filaments. And then when you get to the zone of overlap, you can see that they intermingle. So there's 1, 2, 3, 4, 5, 6 thin filaments for every thick filament. And there's 1, 2, 3 thick filaments for every thin filament. So there is a specific ratio that's achieved, but you can only really see it when you look at the cross-cut section. So once you conceptualize that in order for a muscle contraction to happen, the proteins that make up the thick filament have to reach out and grab onto the thin filaments, and they do that on all sides. That should provide you with enough information to understand that, oh, hey, if you add that up, that's actually a lot of pulling power if you consider it at the molecular level, and then you multiply that out to many, many, many sarcomeres in many, many, many myofibrils in many, many, many muscle cells. That's considerable. That's enough for you to do things like support your own body weight, do a push-up, do a chin-up if you're able. So what these proteins are able to accomplish is pretty impressive considering their relatively simple geometric arrangement. Okay, so thick filaments. I mentioned that they're made of a protein called myosin, and that's true. So each thick filament is made of about 300 myosin molecules wrapped together. So down here, this is a myosin molecule. It has a long straight part that's the tail, and the long straight parts basically bundle up with each other to form the thick filament. And then there's a myosin head, which is the portion that actually grabs the thin filament, and it's also an ATPase, meaning that it cleaves ATP to gain energy to use in muscle contraction. So again, muscle contraction is very expensive energetically. It requires a lot of ATP, and this is the reason why it requires a lot of ATP. Every time this myosin head wants to move into the correct position to grab onto the thin filament, it has to spend 1 ATP. So if you have oodles and oodles of thick filaments in many, many, many sarcomeres, up to 10,000, and many, many muscle fibers in that muscle, or even in that fascicle, the number of ATPs required to achieve muscle contraction starts to get very large very quickly. So that should help you understand why there's so many dang mitochondria there, and also why muscles, striated muscle and skeletal muscle in particular, demand so much blood and oxygen out of your cardiac output. They need it because they need to keep making ATP to keep up with the intense demand of these myosin heads. So these are energized by ATP, meaning that they bind an ATP, they cleave it, and then the little burst of energy that they get from that last phosphate on the ATP breaking off, they use to move into the correct position to grab onto the thin filaments, which are made of a protein called actin. So here's the components of a thin filament. They're more fancy and they have more moving parts than the thick filament, but that's all for a very good reason. And the reasons are related to a couple things. One of those things is grabbing power. The other is the ability to be grabbed. And that's where calcium comes in. All right, so let's take a look at this strand and we'll just build it up piece by piece. So let's zoom in a little bit, if it will permit me, that is. There we go. Okay, so here's our thin filament. As you can see, there's a lot going on here. So notice that bracketed here is the F-actin strand, meaning that F stands for filamentous here. It means that the individual G-actin molecules, so each of these little orange-yellow balls, have polymerized into this double helix that makes the F-actin filament. So the filament is made out of G-actin, if that makes sense. So we call these G-actin molecules or G-actin subunits because they don't ever function all by themselves. They have to be in a strand like this with these other proteins in order for them to work. So that's the difference between G-actin and F-actin. So each of the G-actin molecules has an active site. So you might remember from proteins and protein folding, a protein with an active site is usually an enzyme, sometimes not. But an active site is a place with special shape and special chemistry on the surface of a protein that allows it to interact with another protein. And that's true here as well. However, this rope-like protein called tropomyosin is blocking the active site of each of the G-actin molecules. On the tropomyosin is another protein called troponin. So troponin is where calcium binds. If calcium is released into the sarcoplasm and allowed to diffuse out, it will bind to the troponin, and that moves the troponin and the tropomyosin off of the active site, which makes the active site open, uncovered, and available to be bound by myosin if there's enough ATP present for myosin to do its thing. So that's why we have these golden ingredients that are necessary for muscle contraction, where we need calcium, we need cytosolic calcium to be high, and we need a continual supply of ATP in order for both the actin and the myosin to function correctly so that they can join together. Nebulin is just basically the axis along which the F-actin orient themselves. So it's the central core. It forms the base structure, so to speak. It doesn't actually participate directly in muscle contraction, which we also call cross-bridge formation. It's more of an axis along which all of the other things polymerize, if that makes sense. So, just to review, tropomyosin covers the active sites of G-actin in resting fibers, so fibers that are not actively generating tension. And troponin binds to calcium, which results in tropomyosin moving off of the active site of the G-actin. So that's where the calcium comes in. Repetition is our friend when we're talking about very complex processes like this. So you're going to hear me repeat myself a lot. It's not because I think you're stupid or you can't do this. It's just because, as you're seeing, there's a lot of moving parts to keep track of. And when you're just learning it, it can be really daunting. So repetition helps with that. It helps the information be less daunting. So you're also going to notice this titin molecule that looks like a little green spring. It's not pictured down here because it doesn't participate directly in the formation of a cross bridge between G-actin and myosin heads. What it does is just make sure that in the zone of overlap, the filaments are sliding smoothly past each other and not getting off track or derailed or running into each other. So titin is there for stabilization purposes. So the sliding filament theory is what I just described, where the zone of overlap changes length because the amount of overlap between actin and myosin, between the thin and thick filaments, changes length as well. So the sarcomere gets shorter because the zone of overlap gets larger, the I band gets much smaller, and that's how tension is generated. So it's not that the individual proteins bunch up or get shorter, but rather they slide past each other like interlocking comb teeth, and that results in the shortening of the sarcomere overall, which you can see if you look carefully at this image versus this image. So note, the length of the A band has not changed, but the length of the I band and the H zone has changed. So the I band and the H zone have both gotten shorter as the sarcomere itself has gotten shorter. And you can see titin is just very bunched up here because it's just busy being a spring and keeping everything on track. So that's how the sarcomere shortens. Now, I mentioned previously this requires calcium, and it requires calcium to come in from the outside of the muscle fiber, but also requires that calcium be released from the sarcoplasmic reticulum. So now that begs the question, what makes all this happen? So the signal from a motor neuron, so this is a neuron that's going to travel along one of those spinal nerves that I showed you previously, that signal comes in the form of an action potential, which I describe both in the Khan Academy style videos and in the podcast. So I'd encourage you to listen to and watch both of those to get more detail on action potentials. And the action potential ends at this place called the motor endplate of the neuromuscular junction. So the motor endplate is the flattened area of the sarcolemma that contains all of the receptors for the neurotransmitter that's released from the motor neuron, and that neurotransmitter is acetylcholine. So when acetylcholine crosses this synaptic cleft and binds to the receptors on the motor endplate, that ultimately causes the release of calcium from the sarcoplasmic reticulum, and then the calcium is free to go bind to troponin, move tropomyosin off of the active site, and allow myosin to bind to actin. So let's zoom in on the neuromuscular junction and talk about that. And here are some pretty cool Confocal fluorescent microscopy images up here. Well, this one's just light microscopy, but this is confocal fluorescent. So this is looking through multiple cells at once, and they're dyed with some immunofluorescent dyes, which make them light up neon colors. So you can see that the green are the axons spreading out into axon terminals, and then the motor end plates are these spread out zones where you can see the axon terminals ending, and the pinkish lighting up places are places where there are acetylcholine receptors. And you can see the same information here if you look very, very closely at this light microscopy image down here. So there is one neuromuscular junction per muscle fiber, but it doesn't mean that it's not spread across multiple areas. And each of those has axon terminals, which is just the end of an axon. So the axon is the sending part of a neuron, and the end, terminal just means end. So axon terminal means just the end of an axon. So waiting in the axon terminal are something called synaptic vesicles. And synaptic vesicles are little bubbles of membrane that are full of neurotransmitter that are going to be released out of the end of the axon when an action potential arrives at the end of the axon. So in this case, that neurotransmitter is acetylcholine, and that's the one that's at the neuromuscular junction for skeletal muscle. So we abbreviate this ACH. You need to recognize the word acetylcholine if you see it, but I may abbreviate it ACH as well, so I'm just showing you both here. So the motor end plate is the spot on the sarcolemma, the skeletal muscle membrane, with the receptors for acetylcholine. And as I mentioned in the podcast episode about this, the acetylcholine receptors are chemically gated sodium channels. So when their favorite chemical is bound to them, and their favorite chemical is acetylcholine, they open up and let sodium from the outside of a muscle cell to the inside. Once the impulse from the neuron is over, there is an enzyme called acetylcholinesterase that basically binds to any leftover acetylcholine in the synaptic cleft and acetylcholine that's popping off of the receptors, and it breaks up that acetylcholine into two parts, rendering it useless. And then the acetylcholinesterase basically shoves those two parts back into the axon terminal where they can be recycled and recombined into a new acetylcholine molecule. So we'll talk more about that as well. So the next few slides are just going to zoom in on exactly what's happening during that process. So this is one of those times, this is one of those times where I warned, hey, check out the figures in the textbook before you watch or listen to the lecture, because then when I show you a big scary picture like this, you'll at least have seen it already and it won't be so scary to you. So let's break it down piece by piece because that's how this information is less frightening looking. So at a resting neuromuscular junction, one that's not actively initiating contraction, we've got vesicles full of acetylcholine just waiting. And that's because no action potential is happening in the membrane down here yet. So there's no signal for them to be released into the cleft. We have acetylcholinesterase just waiting in the cleft as well. And then we have the little acetylcholine receptors along the motor end plate of the sarcolemma. And what you're going to notice is that there's this thing called a junctional fold where the sarcolemma bunches up in the area where it's associating with the neuron. That's just a strategy to put more acetylcholine receptors in this area. So unfortunately, they're not illustrating that very well. They really could be doing better about this, but the idea is that if you increase the surface area, you can put more acetylcholine receptors in this spot, and that's going to increase the sensitivity of this spot to acetylcholine. So once an action potential arrives, these little acetylcholine vesicles are going to fuse with the membrane. And they do that because the action potential arriving opens some voltage-gated calcium channels, and that lets calcium in. And the calcium going into the end of the axon is what tells the vesicles to fuse with the neuron membrane and let the acetylcholine out. So that's not pictured here, but it is important. Sorry, I bumped the mic. I hope that wasn't too loud. That's not pictured here, but it is important for understanding chemical synapses, which is what this kind of synapse is. So I'm including that information extra so that you know what actually causes the fusion of these vesicles with the membrane. The action potential allows calcium to enter the end of the axon, and that's what causes the fusion here. So as you can see, the fusion just results in more acetylcholine, formerly having been contained in the vesicles, but now it's out in the synaptic cleft, and it can diffuse across the cleft and bind to the receptors. So when the acetylcholine receptors bind to an acetylcholine, they open up to let sodium through. And sodium is going to depolarize the membrane in this area, and the sodium ions are also going to diffuse laterally to go change the membrane potential in neighboring areas. So until now, this area of the inside of the sarcolemma was very low or poor in sodium. But now that sodium is rushing in, because the concentration gradient is very steep, we're going to suddenly have a lot of sodium in this area, which results in the sodium diffusing laterally. And over here, which is unfortunately not illustrated very well, but I do illustrate it in my drawings of this, over here are some voltage-gated sodium channels, which open in response to a change in membrane potential. So a change in membrane potential, in this case, from the resting potential up to about positive 30. That's going to basically carry the action potential across the cleft and over into the rest of the sarcolemma. And then it rushes down and spreads across the sarcolemma. So if there's only one impulse or one action potential that has come down the motor the motor neuron, and then it stops, there's going to be a cessation of the signal to fuse more vesicles with the membrane. So this action potential is going to go off and release calcium in a way that I'll describe in a little bit. But after that, there's no more signal. So the acetylcholinesterase is going to interact with the acetylcholine that has now popped off of these receptors and break it up and put it back in here for reassembly. So what happens when the action potential spreads along the sarcolemma? Well, I mentioned those T tubules previously. The voltage-gated sodium channels open in response to a change in membrane potential from neighboring voltage-gated sodium channels opening. So it creates this propagating and spreading wave of excitation that is self-reinforcing and self-spreading, and that excitation spreads down the T tubules to the area where the T tubule interacts with the terminal cisterna of the sarcoplasmic reticulum. Specifically what it does, which is really cool, is there's a little protein door or hatch in the sarcoplasmic reticulum that is connected by a connectee protein piece to the T tubule. And it's voltage sensitive. So when the action potential rushes past this little voltage sensitive piece, the hatch pops open and lets the calcium ions into the cytosol. The calcium can then bind to troponin, move the tropomyosin off of the actin's active sites, and assuming there's enough ATP around, the myosin heads can reach up and grab onto the actin active sites and pull back to increase the zone of overlap. So as the thick and thin filaments pull on each other, the sarcomeres shorten, and that leads to the muscle fiber contracting. So we call this tension production because when a muscle is producing tension, there's two options. It can either get shorter, or if it's opposing some equal but opposite force, it can stay the same length but hold in its position. So an example of that would be if you hand me a couple cinder blocks and ask me to hold them. If I'm holding them steady and not setting them down or picking them up, my muscle is not actually changing length, but I'm having to use a lot of ATP to keep my arms in the same position because I'm having to generate enough tension to oppose the weight of the cinder blocks. So we'll talk more about that later, the difference between kinds of contraction. So that's why we say tension production instead of necessarily shortening. So it can be different. So this slide is just a reminder of the myofibril arrangement and the fact that the zone of overlap is the area where the actual shortening happens because the thin and thick filaments are going to slide past each other until there's no more zone of overlap possible. So until the thin filaments are almost running into the M line and the thick filaments are almost running into the Z disc. That's the maximum possible shortness that can be achieved. Okay, so let's look at another view of this. What this is picturing is this situation right here. So we're going to look closely at what's actually happening in this picture. So these little images are a zoomed in version of the relationship that we're seeing here, just to give you some context, because I know it's hard to understand when you're just looking at it. So this is the myosin tail, and then here you're seeing a myosin head and another myosin head. So in the resting state, you'll notice that the myosin heads are not interacting with the actin. There's the space between them. But once the action potential in the T-tubule opens that little hatch and lets calcium out, that's going to bind to the troponin and move the tropomyosin, which is that strand-like one, off of the active site, which is this yellow part here. So once the active site is exposed, because now there's enough calcium to bind to all of those troponins and move all of those tropomyosins, then, assuming that we have ATP, the myosin heads can reach up and grab the active sites of the actin. And they're doing it on all sides. So there's a lot of cross bridges being formed, not just between one thick filament and one thin filament, but between many thick filaments and many thin filaments in the entire zone of overlap. So here's another picture that looks scary at first, but we're going to break it down piece by piece. And once we see that it's just a bunch of moving parts, but if you take it step by step, it's not so bad, this picture becomes a lot less scary. Yes, there's a lot to keep track of, but as you repeat this and practice drawing or explaining it yourself, you'll get better and better at walking yourself through these series of events, and it won't seem so scary by the end of it. So this is where trying to see if you can explain or draw or write this out without looking at the book is going to help you figure out how much of this you really know and what parts of it you're missing as you're trying to explain it to yourself or to somebody else. So that's why I can't recommend enough either taking a blank sheet of paper and trying to diagram all of this out, or trying to explain it to one of your family members who doesn't know anything about this, because they're going to ask clarifying questions. And if you can't answer the clarifying questions, that'll be your cue that, oh, I need to work on this a little bit. Okay, so in panel 1, the contractive cycle begins. So note that in this picture, the myosin head has already cleaved the ATP. So we have adenosine diphosphate, which is 2 phosphates, not 3, and an inorganic phosphate, and this is the phosphate that used to be part of ATP. So the fact that they're separate means that the ATP has already been cleaved. And that's what moved this myosin head into this position where it's kind of jumping up to be closer to the actin, versus this myosin head you can see is resting and it's kind of pivoted down closer to the tails. This one's reaching up. So once that has been energized, we call this the energized state or the cocked state, which is referring to like cocking of a pistol where you pull the hammer back and that readies a gun to fire. Same idea here. So this is the cocked position where the myosin head is energized and it's ready to bind to the actin active site if it's permitted to. So once the calcium comes out of the sarcoplasmic reticulum and enters the sarcoplasm and is able to bind to the troponin, it's going to move the tropomyosin off of the active site, which you can see are these little divots here on the actin. If those two criteria are reached, we've got cleaved ATP and energized active sites, or cleaved ATP and energized myosin heads plus revealed active sites due to the presence of calcium, then we can form a cross bridge, which is when a myosin head grabs on to the actin active site. So in response to this, once the binding is achieved, like with many proteins, the binding of an active site and its substrate, whatever that is, causes a conformational change in the protein, which is a chain reaction of little movements in the amino acids that make up this folded protein that change its overall shape. And in the case of myosin, this conformational change causes two things to happen at once. Thing number one is it lets go of the ADP and inorganic phosphate that it was holding on to. And while it's doing that, it's also bending unidirectionally towards the M line. So the head is pivoting towards the M line, which is going to move the actin and basically cause these filaments to slide past each other. And this sliding is called the power stroke. So this is the actual time and picture of when the sarcomere is getting a little bit incrementally shorter during this pivot. After the pivot, another ATP comes along and it binds to the myosin head, which causes the head to detach. Because the myosin head has let go of its ADP and inorganic phosphate during the power stroke, now that there's another spot where the ATP can bind. And when it does bind, it makes the myosin head let go of the actin. Once the ATP is cleaved again, we can have another cross-bridge cycle. So you'll notice that the ATP being cleaved resets the myosin head into the cocked or energized position, and assuming we still have calcium causing the reveal of the active site of the actin, we can have another cross-bridge cycle. So we can have myosin reach up and grab the active actin again and perform another power stroke. So the cycle continues over and over again as long as there's enough ATP and as long as there are enough calcium ions. So when you are actively contracting your muscle, what you're doing is consuming ATP and consuming calcium until you run out of one or the other. Typically, you run out of ATP first. So we're going to put it all together here and then move on conceptually, and then we can shore up all of this information because, again, I understand that it's a lot, using the YouTube drawing videos that I've already posted and the podcast episodes that I've produced about this very content. And we can also work it out in person on the board in the lab because, again, this is a lot to keep track of. And if you've never thought this tiny before, it's going to be a bit of a challenge. So not to worry, we'll help you through it. I'll work through it with you. We can all do it together. So chemically gated channels in the form of acetylcholine receptors respond to acetylcholine being released from the end of a motor neuron. And that acetylcholine is released in response to an action potential coming down that motor neuron. The acetylcholine binds to these chemically gated channels, and that opens sodium channels and lets sodium in here. The sodium diffuses laterally and opens voltage-gated sodium channels in neighboring areas of the sarcolemma, including the T-tubule, and that propagates the action potential across the sarcolemma, allowing it to spread throughout the sarcolemma, including down the T-tubules. When the action potential makes it down to the T-tubule, it's going to affect that little voltage-sensitive gate, which opens the calcium hatch in the sarcoplasmic reticulum and releases calcium from the sarcoplasmic reticulum. Calcium diffuses out, and calcium binds to the troponin, which moves the tropomyosin off of the actin active site. So after that, as long as we have enough ATP to continually recock the myosin head and allow the myosin head to release the ADP and inorganic phosphate and perform the power stroke, we're going to have enough ingredients, so to speak, to cause the zone of overlap to increase and generate tension in our muscle. So here is a picture, and I'm not going to quiz you on the specifics of this. It's just cool to be able to see it. So here's this little voltage sensor, and this is the little calcium gate. So when an action potential passes this voltage sensor, it basically tugs this little gate open, and that's what lets the calcium out. The gate lets calcium out at a rate that's faster than the calcium is pumped in, and that's what allows calcium to continually diffuse out. So this pump is always on. It doesn't turn off. It's just that if the voltage sensitive hatch is open, calcium is able to diffuse out repeatedly. So it's pretty cool that we have this little voltage sensitive hatch where an electrical potential is changed into the kinetic energy of motion, which pulls on and opens this little hatch in the sarcoplasmic reticulum. Pretty nifty. So what about relaxation? What does it take to relax a muscle? Well, relaxation is passive. All you have to do to relax a skeletal muscle is not send it any more action potentials along that particular motor neuron. You just have to stop doing it. When that happens, the acetylcholinesterase is going to grab that acetylcholine that's left over in the cleft, break the acetyl group off of the choline group, which renders the acetylcholine not able to attach to its receptor any longer, and then the little halves of the acetylcholine get shoved back up into the axon terminus to get recycled and reassembled into new acetylcholine. So that's handy because that means that there's no extra step necessary in order to stop contracting a muscle. And it also means that you can recycle the neurotransmitter and you don't have to make it from scratch every time you want to contract a skeletal muscle because that would cost way too many calories. So the sarcomere lengthens during relaxation because the calcium levels in the sarcoplasm are going down. The little gate in the sarcoplasmic reticulum is no longer open, and that calcium pump and calsequestrin are gathering up the calcium from the sarcoplasm and sticking it back in to both the sarcoplasmic reticulum, but also the extracellular fluid. So there's pumps that move calcium out into the extracellular fluid as well. So if we have calcium levels that are declining, it doesn't matter how much ATP is there because the active site for the actin is blocked, which means that there's no possibility for cross-bridge formation. So that's what I mean when I say relaxation is passive. And here's a nice picture from your book that basically compares and contrasts the two states where we're initiating muscle contraction and then we're allowing the muscle contraction to stop. So I'm not going to read these to you because I've just spent a lot of time explaining both sides, but I would encourage you to just review these because it'll help you visually compare the two states and how they're different, which will help you understand the difference between active contraction and muscle relaxation, and also reinforce the idea that if you want to initiate and then sustain contraction, so have contraction keep going, that means that you have to keep sending impulses from the central nervous system down this motor neuron. So you don't get to just activate the muscle once and then you're done and it stays contracted. You have to continually bother it to stay contracted. And that is the end of part 1. So I'm going to render this really quickly and upload it, and then I will get right to work on part 2, so you'll have both of them by the end of today. Thank you for your attention, and I will see you very shortly in the next video.